Okay, so we've covered a lot of stuff so far today, and I'm only going to talk, looks like maybe four more slides. Four more slides today. We had said that, again, the graded potential, in terms of voltage, as long as it doesn't reach minus 55, it can go all over the place. And so you can open, depending on the neurons that are communicating together, you can be opening different kinds of chemical gated channels that are making it either more polar, hyperpolarizing, and that's if you open a negative ion channel, or depolarizing, and that's if you're opening a positive ion channel, such as the sodium gated channels. So these chemical gated channels with the graded potential, there's one of my slides, I call it a local potential, but and why am I calling it that? Well, it ends up, remember our nerve, our neuron, we have our neuron, and we have, I almost dropped my stuff, sorry. So you can see we're going to talk about how we have partnered, we're going to have, what did we say? Spit it out, Steve. What did we say is the role of the dendrite? The dendrite is where we receive information, right? Those things extending up on the top. And so what that means is you're opening chemical gated channels there. And say we're opening sodium chemical gated channels up there on those dendrites. But you notice that they're up on the other side of the soma, the cell body, from the axon hillock. So when we talk about graded potentials, they're different at different locations on the nerve. But we had said there's only the most important location on the nerve for threshold is going to be the axon hillock. And why is that? It's because it is the trigger zone. It's where we generate an action potential. So the local potentials can vary, and you can have even different millivolts in different parts of the neuron. But what's important is the potential at the axon hillock. So we had said those local potentials, they're highly variable. They can go either direction. And then we had said, in contrast to that, the action potential, that's a nerve impulse, we had said it's generated at the axon hillock, and it results in this spread of in, sodium in, potassium out, sodium in, potassium out, sodium in, potassium out, as it goes down the neuron. Okay. So remember we were talking, when you looked at this nerve, I keep showing this picture of this nerve. Where is this baby? Remember we've been looking at this nerve, and look at these things. What are these right here? If you could look carefully, you see what it says, a Schwann cell. What did we say? We said Schwann cells are little insulating cells that wrap around the axon. And we had said that what they do is they create a faster action potential. So if you look in between those, there is that right there. It says a node of Ranvier. Did you ever watch the Addams Family? I know, I'm digressing. The Addams Family, Morticia and, what the heck was the guy? Morticia and, I don't know what, the guy and the girl, I can't remember their names now. But the guy would get really excited when the woman spoke French to him. And nodes of Ranvier, that's French. So anyway, it's just one of these weird thoughts that come into my head occasionally. Anyway, the nodes of Ranvier are very important. So if you don't have the places where you've covered up all of the surface of the axon, you have continuous conduction. And that's what I described. In, out, in, out, in, out, in, out, all the way down. But if you have Schwann cells or oligodendrites in central nervous system, they also have nodes of Ranvier between them. These are little openings where the nerve impulse jumps. So we had said, what is a nerve impulse? We had said it's depolarization, repolarization, and then refractory period. It happens at one node, and then it jumps to the next node, and it jumps to the next node. It's much faster. You don't have to go through all of that cell membrane. So continuous conduction is if you don't have oligodendrites, or if the Schwann cells aren't fully wrapping the axon in the peripheral nervous system. If we are talking about, but then if you have those, what do we call that? It has its own name. It is called saltatory conduction. Saltatory conduction is this jumping from node to node. And so here we can see the nodes of Ranvier, and what's going to happen is the impulse is going to jump from node to node, and it's a much faster form of action potential. Okay. So we have saltatory conduction. And then we had said that what is the point of a nerve impulse? The point of the nerve impulse is to secrete that neurotransmitter substance, right? So where do you secrete it? You secrete it between the presynaptic neuron and the postsynaptic neuron. So before, when I was talking about local potentials, we were the postsynaptic neuron, right? We were the one that was being affected by our partners. As soon as we send an action potential, we become the presynaptic neuron. Why? Because the name, we're talking about the synapse. And a synapse is a little teeny space, literally like 20 nanometers, very, very small space between a presynaptic neuron and a postsynaptic neuron. And since we had an action potential, we are the presynaptic neuron, and that means we are going to be secreting the neurotransmitter substance into the synapse, and it's going to bubble across to the postsynaptic neuron. And what's it going to do? Think. It's going to open chemical gated channels and cause, what do we call it? A graded potential on that next guy. So we have presynaptic neurons and postsynaptic neurons. And in the vast majority of nerves, we're going to talk about release of a neurotransmitter substance. There are some that we just mention here, and we'll never come back to them, that have what we call electrical synapses, and that's where the action potential jumps basically across to the, instead of jumping from node to node, it jumps across to the next neuron and causes an action potential that way. It's a much rarer, and it's only found in the central nervous system. All right, so one last slide, speed of transmission. The speed of the action potential. Now, I'd said before, sometimes, remember in the very first lecture, I had talked about neurons. And I'd said, if you are a large peripheral nervous system neuron, we had said the Schwann cell wraps around your axon. And then we had said, but we have some that are small. So what are we meaning? We mean physical size. We truly are meaning the physical size of the neuron. So the diameter of the axon will have an effect on the speed of the nerve transmission. So we have, what factors affect the speed of the transmission? The size of the axon, whether or not you have myelinated cells. Now, myelin is the material, the insulating material that is in a Schwann cell and also that is in an oligodendrite. So when we talk about myelinated neurons, that means they either have Schwann cells or oligodendrites wrapping around their axons. Also, temperature. Now, inside of you and I, how much does our temperature fluctuate? And you say, well, you can find that out by taking your temperature, right? So chances are it doesn't fluctuate a whole lot, right? You say, yeah, well, my temperature is 98.6. I'm normal. And maybe you got a little bit of fever and you're 1 degree hotter. I'm 99 and I don't feel so good. So your temperature doesn't affect much. It doesn't fluctuate much. So in you and I, the speed of the transmission, while it would be affected by temperature, it really isn't in practicality an issue. Why do we talk about it? You know what the animal is? The animal model for studying nerves, and the reason that you study them using squids. Squids and octopi, they have giant neurons. And so if you're studying nerves and nerve transmission, you say, well, it's a heck of a lot easier to work with a nerve when it's big. So most of the neurophysiology, this stuff we've been talking about today, most of this neurophysiology has been studied in squids. They're cold-blooded. So if they're in hot water or cold water, we know the effect that it has on nerve transmission. Hot water, it goes faster. Cold water, it goes slower. All right, so that's all I'm going to talk about for today. This is a pretty heavy-duty kind of a lecture, so we're going to stop right here with speed of transmission. You guys have a good day. I'm going to get this stuff uploaded to my partner YouTube, and then I'll get it posted. All right, talk to you later.